Okay, so it's 15 past. So welcome everyone to this machine learning coffee seminar. So this Monday we have Professor Leo Lahti from the University of Turku, who is going to talk about probabilistic machine learning and human microbiome research. And if you have any questions during the speech, just go ahead and write them in the chat. Okay, please, Leo.
Okay, thank you very much, and thanks for this invitation to present our research. It's kind of funny because I did my PhD in one of the FKI member labs, and I remember, if I remember correctly, we were at that time launching this coffee seminar. We saw Nature journal laying on the coffee room, and there was a story about coffee seminars in some other lab, and we thought if we could have the same. And initially it was just like a 10-minute, very brief paper presentation during the coffee break at 2 o'clock in the afternoon. And nice to see that it has evolved into a proper seminar series. I have been, after the PhD studies, I have been focusing mainly on the human microbiome studies for the last decade. It's a really vast topic, also in terms of machine learning aspects, and I definitely won't cover all of that, but I wanted to give some examples about how probabilistic machine learning is being applied in this field. And before we go into that, just some background. So our planet has been inhabited by microbial life since the very beginning. The earliest records are somewhere in 4 billion years ago, and these were the first life forms that existed on Earth. So all the higher organisms have evolved to live in symbiosis with microbial organisms. And these ecosystems of microbes are not only necessary and important for our health, but also for the planetary ecosystems and many other things. And the modern science on microbial life started somewhere in the 17th century after Antonie van Leeuwenhoek invented the microscope in Holland. And he started to observe these small beings in different kinds of body fluids, for instance. So this pioneering work has then led to many subsequent research. And the human microbiome is about 1 to 2 kilograms in weight, and it contains actually more microbial cells than our own body contains our own cells. And the gene pool is even larger. So the microbes that live in our body, their metagenomic gene pool is 100 times larger than our own gene pool. They have millions of genes in the gene pool, whereas our own body has approximately 20,000 genes, human genes. So this gives just some kind of idea about the genetic potential and importance of these microbial ecosystems for our own body. And they are, of course, in constant interaction with our body. We need a right balance of microbial species. And these are ecosystems. They have potentially alternative stable states, as we will see. And this balance between pathogenic state and healthy state can be potentially tipped through external perturbations, for instance. And then, of course, we need to measure these systems. So if we want to measure the gut microbiome, for instance, gut microbiome is one of the most diverse microbial ecosystems in the human body. If we want to measure this, one very handy way to measure is the fecal samples, because this is non-invasive method and we can find volunteers easily. And then, of course, we need to record all kinds of background variables like stool consistency, medication, dietary habits, lifestyle, and so forth, and take these into account into analysis as well. And the current analysis has been moving very much from individual bacterial or viral species to ecosystems of organisms. And And so these are highly multivariate systems where we want to understand the overall dynamics on the system behavior. And also how to manipulate these systems. That's why we need to understand their composition and dynamics, because we potentially want to manipulate these ecosystems, for instance, to recover health. And one very drastic measure for this is fecal transplants that have been used to recover people from severe antibiotic diarrhea. And just a couple of days ago, our colleagues from the University of Helsinki published this paper in Cell about maternal fecal microbiota transplantation in cesarean-born infants. So there are many applications emerging with important biomedical applications. But then, of course, a word of warning, there is also a lot of do-it-yourself kind of fecal transplant video material in the internet, so heavily not recommended to yourself. This can be potentially fatal if not properly tested for pathogens. Okay, so the cultivation-based techniques have been traditionally used to measure individual bacteria, but now the DNA sequencing has revolutionized the whole field. So we can identify microbial organisms based on their microbial sequences. So metagenomic sequencing, shotgun sequencing, is being very actively now used in this field to measure the overall metagenomic content in different microbiome samples. And then one of the first problems in understanding the microbiome is how to assemble these metagenomic reads into actual metagenomic species, so that we can get a complete picture about the content of the taxonomic groups in the samples. So context clustering is one part of this. So we, if we initially have the DNA sequence, in typical metagenomic sequencing nowadays, it is being split into much shorter sequences in the measurement process, and these need to be assembled by computational means in longer ones. And we call these contigs. And often the contigs themselves are not yet overlapping, and we would need to know which contigs come from the same species. We can do clustering for the contigs, for instance, by analyzing their co-abundance patterns across the different measurement samples. And then we can reach clustering for the contigs. But there is also other type of information available. So there is the DNA sequence composition of the contigs, for instance. And if we look at different stretches of sequences, we can see that different metagenomic species may have different profiles across the DNA composition. So often we can look into K-mers, for instance, 4 base pair strings along the sequence. So there are 136 potential tetramers with 4 base pairs available. And each metagenomic species has a peculiar distribution over the different tetramers, how often these occur along the DNA sequence of that metagenomic species. So we can also construct the tetramer profile and use this also for the clustering. And in one earlier study, what we did was to combine the co-abundance information with the sequence composition information. And this gives us a relatively high dimensional profile over these features, and we can then use this to cluster the metagenomic, the contigs into metagenomic species. But then there are quite many technicalities involved, and actually the use of this coverage plus composition feature vector as such does not really work. It gets a bit too high dimensional for practical clustering purposes. So we do some denoising with PCA and drop the dimensionality to essential 90% of the information content before we apply the multivariate Gaussian clustering. By this, we can actually improve the clustering results in the validation data. And then the multivariate Gaussian clustering is being used, but we use non-parametric mixtures because we do not really know the correct number of metagenomic species in the samples. And we were using variational Dirichlet process multivariate mixtures for this infinite mixture modeling, and it seems to work very well. We initially were trying more traditional methods to fit the mixtures, but the VDP led to at least 100% speed up, and also more accurate results, which we were slightly surprised at first, but it seems to avoid local minima so nicely that that probably is the explanation for the better accuracy. And in this figure, you can see real validation of this in a mock community. So the different ellipsoids are here, clusters identified by this method, and the different colors correspond to real known metagenomic species in this mock community. And this was joint work with Christopher Quince, who has been my good colleague in these studies for quite some time. So this is just one example about the pre-processing problems that come across in analyzing microbial data. So this is before we actually even get to the downstream analysis of analyzing and understanding the ecosystems. And typical outcome from this kind of pre-processing tasks in microbial community analysis is the taxonomic abundance table, where we have taxonomic groups on the rows and then individual samples on the columns. Yeah, if some of you might be familiar with gene expression matrices, so this is just replacing the RNA transcripts with the bacterial groups. And each individual has a quite unique fingerprint over the microbial taxa, and then we can also see that certain taxonomic groups frequently co-occur together across the different individuals. Red denotes high amounts of certain bacteria in the sample, and blue denotes low levels. And in analyzing these systems, we can, for instance, approach this by applying all kinds of latent variable models. So in this field, we also use the typical latent variable models like probabilistic principal component analysis, non-negative matrix factorization, CCA, other models to understand these data sets. A very nice review of these models in this field is given by Susan Holmes in this paper that you see on the screen. So now then, once we have collected this kind of data, we want to understand the overall population variations. And in Finland, we have this FinRisk data collection that is 7,000 individuals from different parts of the country. And this ordination plot, this is multidimensional scaling, which we in this field call principal coordinates analysis for some reason. And each point on this map is one individual, and the similarity of the individuals is now based on the bacterial abundance profiles. And as you can see, there is at least two clusters on this map, the green cluster and the red dominated cluster. And these correspond to individuals that have high levels of certain bug Prevotella. The green individuals have very high levels of Prevotella, and the other cluster has very low levels of Prevotella, which is one of the important bugs in our gut ecosystem. So there is certain structure in the population variation, and we want to understand these structures. And one of the applications for the latent variable models in this field has been the community typing. So, or we call it also a landscape model. So we want to understand if there are latent components or latent community types that could explain this individual variation. So individuals might potentially cluster into different types based on the microbial composition. And there has been different ways in the field to approach this. And one problem with this is that microbial abundances are most of the time compositional. So this means that we don't really have access to the absolute abundances of the microbes in the samples. We only have relative abundances with some up to 100%. And this limits the availability of the different methodologies to understand this. So multinomial sampling is a handy model for such compositional data because we essentially require the probability is sum up to 100%. So we have been often using multinomial as the observation model for compositional microbiome data. So in this case, X or the observation is the microbial species counts from the sample or the sequence counts. And then P, which is the parameter of the model, is basically the underlying true abundance of the different microbes. And then we have K different microbial taxa in this sample. So there's the true abundance P and then the observed abundance X. And we use multinomial to model that. And Dirichlet multinomial is often being used because the Dirichlet gives a nice conjugate prior for the multinomial, and then we can have a little bit more flexibility in this model and marginalize the true unknown species counts or species proportions out from the model. In this context, the alpha parameter of the Dirichlet multinomial is called a so-called biodiversity parameter, which regulates on how evenly the different species are distributed. So we could start with that. But if we speak about community typing and we consider the potential for multiple community types, other types of models have been suggested, and the Dirichlet multinomial mixture is one example about the model where the Dirichlet prior for the multinomial is replaced by a mixture of Dirichlets. So we instead have K different Dirichlet components, and then the model is such that each individual is assumed to come from one of these components. So we can have 3 or 4 or more community types. Each community type is tied to one of the Dirichlet components, and then each individual is pulled out from one of these components. And then we can, of course, do some validation on how many clusters there are, learning the number of mixture components. And this has been one of the very popular models in the field for determining the numbers of community types. This has some problems, like the Dirichlet model itself has problems that it does not take into account the synteny between the species. For instance, synteny means that certain species naturally tend to co-occur together. But the problem with the Dirichlet is that once one species gets more abundant in this model, it means that all the other species will decrease in abundance because the probability is sum up to 1. So this kind of synteny effects are not properly taken into account by the Dirichlet model. Then there are questions about the different modeling assumptions, and also surprisingly, the fact that the number of clusters actually depends on sample size when we start to fit these models. And the problem is that these are often being fit by researchers who necessarily don't have full understanding of all the properties of these clustering models in machine learning. And if we use non-parametric clustering, when we have more data, we can always have more structure in the model. So this has led to some confusion in the field on for instance, regarding the correct number of clusters in this kind of samplings. Another problem is that this model and some other models focus very much on ecosystem level clustering. So we really look at the whole ecosystem as one bunch. And of course, it's then clear that we can only see very broad community types or very broad clusters of the community, which is sometimes important because we can attach this to some health aspects in some cases. The community type or part of the community where you're located in might be related to health conditions, and if we can modify this, it may have important consequences. Maybe it is more important to focus on broad ecosystemic patterns, for instance, based on fecal transplants. However, the sub-ecosystem level analysis is more and more coming. We have quite a lot of evidence that the sub-ecosystems are perhaps at least as important as the whole ecosystem, and the actual state of the whole ecosystem can be represented as a rich mixture or rich combination of sub-ecosystems that vary relatively independently. We have been publishing some material on this. But then, if we want to understand this whole biodiversity, we need to go back to the ecological models of biodiversity and understand what is the origins of this variation. Of course, in humans, in the human host, we have a lot of host regulation going on. So human body is all the time regulating these microbial ecosystems. But we also need to understand the natural ecological properties. And the neutral model of evolution is a null model for ecological communities. And it goes against this traditional view of competition between species that started already with Darwin. So Darwin's hypothesis was that the different species compete from the living space, and this will ultimately determine the community composition. But then much more recently, Hubble has proposed that much of the variation that is being observed in natural ecosystems is neutral. And in neutrality, we basically have assumption that there is no competition between the species. All the variation is due to stochastic variations. And quite recently, we showed with Chris Quince and others that the neutral variation or the neutral model of biodiversity is equivalent with certain assumptions to the hierarchical Dirichlet process. I will explain intuitively why. So basically in this model, we still use this multinomial observation model for the data as before. And but this time we replace the Dirichlet prior with the Dirichlet process, and then we add another level of hierarchy. And then this is being used to model the compositional community data. And so to give some motivation why and how, so if we look at the neutral model, we start with the fixed community of J individuals from two different species. So the black and white balls here. And in the neutral model, some number of species will die, and then they become replaced by the same species from the same ecosystem. And now the species will replicate to fill in the empty positions. And those species that are more abundant initially will be a bit more likely to fill in these positions, because the empty positions will be filled in based on the proportions of the different species in the existing systems. So more abundant species replicate a bit faster. But this is just because of the abundance, not because of some competitive relation. And so it's completely stochastic. So this is the neutral model. One other aspect that we add to the neutral model is the possibility of immigration. So occasionally we can have immigration in the system from meta-community. So for instance, in human gut system, the meta-community is all the other individuals around us, and then potential for acquiring new species from this meta-community. And this adds some richness in the overall dynamics of the community development. And so now, if we think of this model in terms of the multinomial model, so basically we would sample these white and black balls from the multinomial, which has certain probability for the black and white balls or some large number of species. But now initially if we think about human gut ecosystem, for instance, we don't really know how many species there are. So we can't really fix the number of species in advance. And this is why the Dirichlet process prior is quite handy here, because it provides us with this infinite model where we can have a potentially infinite number of different species in the system. And then we don't have to fix the number of colors for the poles in advance. And then we can have this immigration from the meta community as well. And so this is giving this simple Dirichlet process level is giving us the chance to have immigration from the meta community and unknown number of species potentially. But then why do we add the other level of the Dirichlet prior to get the hierarchical Dirichlet process? Is that we also want to allow for the possibility of evolution in the meta community. So also the meta community itself is not fixed, but the meta community can acquire new species by evolution. So there is the phenomenon of evolution at the meta community level and the phenomenon of immigration in the local community level. And we model both of these with the Dirichlet process and ultimately use the multinomial as the observation model. So it's a very natural and nice fit. And it turns out that this model is actually, this fundamental neutral model of biodiversity is actually equivalent to hierarchical Dirichlet process. And this is extremely handy for the studies in ecological field, because there have been traditionally many different complications in fitting this neutral model. And there have been variations on how we can fit this for single meta community versus local community model. But scalable fitting for multiple local communities, for instance, has been challenging, and there have been all kinds of approximation being developed. But now for the HDP models, we have the whole machinery of how to fit them more efficiently, and this provides new, very well scalable methods for fitting these models for real ecological communities. So this is quite recent development in the field, although the models themselves have been known for quite some time. But now, of course, all of this has been mainly based on cross-sectional static data. And if we look at this landscape model of the microbiome and the microbial community types, we would also like to understand how individuals travel along these community types. This is a vaginal microbiome example, and we also have, in this body site, we have different community types, but then it's typical that individuals travel over these community types, for instance, during the menstrual cycle. The black trajectory in this figure is one individual over time. And the one problem with the neutral model is that it doesn't really model the community types actually. So we don't really get these alternative community types out of the neutral model. That's one of the limitations of it. So many interaction models yield more complex dynamics. The problem is just that often these traditional interaction models that we can use require more data than we typically have available in human microbiome studies. But we can do simple analysis based on theoretical models in this case, at least. One other limitation is that the access to mechanistic models in the first place is sometimes a bit too much. I mean, the human gut ecosystem, for instance, is so complex that the single mechanistic models that could describe all the system fluctuations are simply not available. We don't have such model, and any traditional models are way too simplified. So one other option is to try and characterize the basic ecosystemic properties, like higher level properties, like overall resilience, how rapidly, how easily the system will aim to return to its original state, for instance, or how stable the states are. how easily they switch to another stable state. We can quantify many of these things even without access to the actual mechanistic model behind the observations. And Ornstein-Uhlenbeck process is one model that we have been using for this purpose. Again, one problem is that often in human studies, it's very difficult to collect very long and very dense time series, because essentially we would need individuals who collect, for instance, fecal samples every day for a year or something. So this is not often very feasible. And in many studies, we have instead many short time series available. And if we want to understand the overall universal dynamics of the system, we may want to aggregate information across the different individuals. We have recently been aiming to do some work on hierarchical Ornstein-Uhlenbeck processes to aggregate information across the individuals or their time series. This is joint work with Ville Laitinen, who is a PhD student in my group. So we have many different models in this space with interactions or with no interactions. That's the neutral model. And we can have deterministic and stochastic fluctuations. And there are many different types of models that are being applied in this field, and that go somewhere on this overall map. And we don't necessarily know which models to start with. But we recently noticed that the spectrum of the time series may give hints on what ecological processes might be the most likely explanations of the underlying variation. So we have different alternatives. We have, for instance, the neutral model that I explained, or we can have these species interaction models like the classical generalized Lotka-Volterra equations, or we can have other types of community models. And we found out that these different models develop different types of spectrum in the time series. And we can use this to identify which are the potential ecological processes underlying our observed data. So this is a recent paper if you like to have a closer look. Okay. So this is just the last slide because the time is getting over. So real communities are complex mixtures of ecological processes, and that's one of the challenges for these studies. We don't really have access to the full underlying models, and there is a lot of individual variation, like the community types, and a lot of stochasticity or fluctuations that we don't really understand properly. And this is why we need flexible models that fit to the data, which explains why machine learning models are often quite handy in this field. And stochasticity definitely has to be taken into account. And in many cases, even if we don't have access to the mechanistic models, we can still quantify many key parameters like resilience or stability or switches between the alternative states. Okay, so of course, I like to thank my collaborators who made this research possible by providing a lot of data, very good data sets for this research, and very good ideas also in terms of the models. And for some of the work that I discussed, you can find links to the publications from our research website, and feel free to ping me in social media as well. So now, if there are any questions or discussion points, I am more than happy to take questions and discussion. Thank you.
Yes, thank you, Leo, for the really interesting talk. So any questions? Yes, Sami, go ahead.
Hi, Leo. Thanks. Very nice talk. So what kinds of data amounts, sample counts are we talking about in the data sets? So why I'm asking is that is there, what kinds of opportunities are you thinking that there would be to apply the deep learning type of models or other types of models that would require more data? And then the other question is that is there data sets where you would have additionally covariates of the individuals, so that the goal would be to either to predict or analyze the relationships with the individuals, which I suppose is one of the main goals, of course, ultimately of these studies.
Right. Yeah, thank you. The first question, the sample sizes vary a lot. I mean, we have all this variation from very small case control studies to large cohort studies. And the largest studies, if we look at the higher end, the largest population level studies that exist currently are somewhere around 10,000 samples, maybe a little bit less. That's pretty much the upper limit where we get some thousands of samples. This Finnish cohort that I mentioned, the FinRisk, is actually one of the biggest cohorts with 7,000 individuals. The one limitation is that these are often cross-sectional data sets only, so we have just the one sample per individual, so we don't really get to understand the dynamics of the communities. And if we want to understand this at the population level, it is of course quite a limitation for the research if we only have one sample per individual and no time series. But you can understand that if the field is limited so that the largest cross-sectional data sets with one sample per individual are around 7,000 individuals, for instance, then taking a second time point would then duplicate this number. And if we want to collect proper time series, we need to multiply this by the number of time points. And currently the cost structure of these measurements is basically limiting very much the population level studies of the time series. So that's the limitation. But in the population level studies, there are already some applications of the deep learning, and that is of course very promising approach, because there is so much variation that is very difficult to understand, and we could potentially mine a lot of different interactions from this data that are not obvious. And for this second question, now I'm thinking what was the, yeah, right. The recording of the metadata or phenotype data from the individuals. Basically, that is of course an essential aspect in all these studies, but it is also very much of a challenge for the field, because many of these cohorts were collected at a time when it was not entirely clear what background material we need to collect. For instance, the consistency of the fecal samples, the liquid content in the fecal samples, is one variable that may influence the observed community profile a lot, and it has been not controlled in many of these studies. The other thing is that in many countries we have quite a lot of limitations on what kind of population level data is available. So in Finland we have very comprehensive population registers, and this is a very nice aspect on actually having a Finnish cohort available, the Finnish cohort, because then we have access to all this Finnish health register data that basically contains data on the medication, all the medication purchases by the individuals, all the doctoral visits and potential diseases. And one aspect that I did not mention at all is that in this FinRisk data collection, it was measured already in 2002, so it's also a prospective data set. So we are not only looking at associations between the microbial community profiles in a single time point with these individuals and their health status, but we can also look to the future, like 15 years after the sample profiling, and we can do survival modeling. And this is of course also one application for the Bayesian analysis. We can do survival analysis to mine which microbial features can predict the future health status, and we have a fresh preprint out of this if you are interested in just now in review. So I hope this clarifies at least some of those questions.
Yeah, it did. Thanks. Very comprehensive answer. Yes. So it seems like a really nice resource also for the methods, people who are developing the methods. But thanks. Antti seems to have a question.
Okay, thanks, Leo, for the nice talk. Really interesting stuff. I was wondering, I mean, from kind of the impression I got here is that, I mean, most of this is basically based on counting the abundance at a pretty high level of phylogeny.
Mm-hmm.
So do you know if there's any activity in sort of linking this more towards what these bugs are actually doing, not just sort of counting categories?
Yes, and this was what I was referring in the beginning, because I wanted to keep this kind of coherent, so I decided to focus a little bit on a certain area of this research, which was just the taxonomic abundance variations. But then actually just counting which bacteria we have is of course just one aspect of this research, and then the other aspect is what these bacteria actually do. And in many studies, the functional products of the bacteria, the metabolite products, are also being measured, and we can get more information about what they actually do. And then if we want to understand together the variation in the bacterial abundances and their metabolite products, for instance, there have been applications of CCA type models, for instance, the canonical correlation analysis type of models, and other multi-view models to understand these links. I think there have been also some applications of the deep learning to understand the connections between the taxonomic abundances and the metabolite products, although we have not been working on that specifically. And the whole field is getting more and more into the actual analysis of the metabolite products. That is typically much better conserved than the taxonomic abundances, and hence easier to model in many cases, and it's also functionally more important.
Okay, thanks.
Okay, do we have any other questions? Okay, so I think we've almost run out of time. So if there's no other questions, I'd like to thank the speaker and also thank the audience for joining. And we will continue next week. So have a nice day.